right. I think you can hear me. I hope you can hear me. Chat at me if you can't hear me. And I know you can see the chat, which is awkward, but here we go with only one desktop. Welcome to Proteus TV. Great. Hi, Santosh. Oh, I'm so excited that you're here, Santosh. Welcome to Proteus TV, episode now. My name is Julie Hayes. I'm a PhD student at the University of New Mexico, and I'm going to talk with you about Season 2, Episode 3. Thank you. Welcome to Season 2, Episode 3 of Proteus TV. And we're going to talk about liquid brains today. And don't worry, you probably don't know what that is. I'm going to tell you. It's very exciting. It's very fun. And we're going to just take a walk. It's kind of like a book report and kind of like a walk down this diagram right here. Just take a walk. So this idea of a liquid brain came about when these people got together a few years ago and just started talking about it. And they came up with this pretty cool framework of what is a liquid brain. Yes, brains. And the framework is right here, what we see right here. So this solid brain is the thing in your head, and what we, classically think of when people say brain. And it's solid because I should back up and say, what are we defining here as a brain, besides the things that zombies eat? Brain is a collection of neural agents that collectively together process information. So it's, we sort of take the world directly from neurons, and then apply it to other things as well. But so this right here is your classic brains, and including computer architectures, right, which are solid. Thankfully, all of the silicon that you're watching me on and I'm coming to you on doesn't move, and that helps it do what it needs to do. Physically, it doesn't, but it's dynamic, in the signals that go across it. And so it's there's two key axes here in this figure that define everything. So we have solid here. The top part is solid in the physical space, and then we have liquid on the bottom. And then the other distinction is between these neural and aneural systems. So we'll get to both of those distinctions. We'll start with this physical distinction, though. So solid, the parts don't move. Liquid. Well, I just did air quotes, but I actually don't think that I figured out how to get my camera on here as well, so I don't think you can see me. But anyway, air quotes, liquid. Air quotes, brains. And so liquid being the agents move. So these here are ants. This is our sort of classical canonical way to think about liquid brains as sort of ant colonies. But these little guys are kilobot robots, and lots of cool work has been done with their architectures, them as, liquid neural brains. And then these are aneural liquid brains. So this right here is Physarum, which is a slime mold, and then these are some sorts of, I don't know, single cellular. This is bacteria, and I don't know what this is, frankly, but it's aneural. So these are agents, cells that don't have as solid of an information processing architecture. And if that sounds fuzzy, it's because it is a little fuzzy, and we're gonna dive into the fuzziness. All right, here we go. Any questions about liquid, solid, neural, aneural before we go forward? For because I can see the chat. All right, so in thinking about, we'll start at the beginning and think, and I said, top to bottom, here we go. And thinking about, our classic solid neural brains, we have this really cool tool that's been developed in the last couple decades called the Connectome, and it is now, as the authors of the sort of standard, the dominant view in neurosciences, and it's pretty powerful. So in case you haven't heard of a connectome, what is it? Very broadly and high level, we're not going to get into the details, but it's another revolutionary omics. And what is omics? Omics, biologists use this sort of term omics, more air quotes that you can't see, omics, to talk about, so things like genomics, proteomics, metabolomics, all of these things where you have sort of an end-to-end information processing architecture, and so if you think about genomics from the genome all the way to a protein, what's happening to make that happen. So they characterize a functional connect. These are my sort of high-level definitions, and Connectomics characterizes functional connectivity of brain regions, literally of neurons, so literally neural, and it's just very, very powerful. The ways we've been able to understand what brains do and how they do it because of Connectomics is sort of revolutionary. And the ways that you might be familiar with the revolutionary nature of genomics, right? The things that we were able to understand about how biology works and evolution happens, and so many things because of genomics. It's on the similar order of magnitude what we understand about solid neural brains from Connectomics. So it's very cool, very powerful, and sort of well known, right? Ish, or well developed as a science at this point. So moving down the our framework here, we get to some great work by fellow Proteans, fellow Institute members, Santosh and Mike, and they have this great paper with a bit of a boring name. Don't let that fool you. It is a wonderful paper where they give us a lens to think about the fuzziness between, right, this neural and aneural, what's the difference, and how to go about thinking a little, what a framework for thinking differently about neural versus aneural systems. And they, we're going to just go through with the frameworks here. This cognitive lens they propose, which I find very useful, I'm just going to take and run with. This cognitive level information processing view of the functions of living systems. This is from their abstract. They get very excited about it and make some sweeping, wonderful claims that I thoroughly agree with. Exploration of the deep parallels across diverse quantitative parallels will drive integrative advances in evolutionary biology, regenerative medicine, synthetic bioengineering, cognitive neuroscience, and artificial intelligence. So that's like pretty big deal, right? That if we can look at systems through this lens, we could really understand it. We can really understand the system on a different level from a sort of information processing perspective that can be really useful. This is a great question. Please interrupt with all the questions. This is meant to not be, I know it's feeling kind of lectury, but I've got the chat up. So, so this is a great question that I'm going to get into more a little bit later. Right, animals are mostly water, so all aren't brains liquid. So yes, and these are great questions. It's a good. I can talk about the distinction now, but I don't know if brains have less water content than other parts of the body. I think that they might. I think neurons have less water content than other types of cells, which would collectively make that true for the brain. But I don't know. Santosh might know. Santosh, if you know, feel free to answer in chat. But this framework of liquid-solid has to do with the movement of the agents among themselves. So, like, whereas ants can change their physical orientation toward each other in space and time, neurons cannot do that. And so it's a little bit of it's maybe not the exact right framework, like phrasing for the framework, but because yeah, I'll get to, like, they're not completely liquid, right? I'll show you exactly what I mean by that. But when we assume they are like fully liquid in the sort of agent sense, we don't get the full picture yet. So stay tuned and keep that question in the front of our minds. It's okay, cool. So and then Santosh and Mike asked this really useful question that I'm going to also take and run with. So again, and they're thinking about remember, they're thinking about solid aneural brains. So that's like what are solid aneural brains that they think about? Like, how does a frog make its body, like the whole rest of the body, not just the brain? Like, how do the cells in the frog come together to make a frog liver or whatever? That's a very kind of gross, or I mean, gross and not quite accurate enough, but example of what we're talking about by these solid aneural brains. And in it's something, so they call that process, right? This idea of aneural cells coming together to do something, basal cognition. And then they ask this question of like, well, it's again, this is this fuzzy area of how do we really know, like, where do we draw the line between cognitive and basal cognition? Like, where is this, or to just use their question, which is well phrased, when does it make sense to analyze a system as a cognitive system as opposed to, like, for them, an exclusively biochemical perspective? So if we're talking about, so just to be clear with the, if this is where we're at sort of exclusively biochemical, but then when does it make sense to think about them as neurons, right? To think about it as cognitively. And the difference here is sort of a top-down, bottom-up idea, that if we're looking at the cognitive lens, we're thinking that there is sort of an emergent property has been adopted by the system and is functioning in a top-down way to affect all the agents in the system, versus bottom-up. Yes, liquid brains are meant metaphorically. Sorry, I was not, maybe you said that recently, or anyway. So, and they give sort of, again, a framework with four things, but they give a little bit of clarity on the fuzziness of when we were going to think about things cognitively versus not cognitively. And cognitive, define cognition as a process in these biological systems when we can say the process is informational, dynamical, statistical, or a learning process. And the other great news is that, like, we don't really want to, we can sort of leave it as kind of fuzzy as far as what we think of as cognition and not cognition, and I'm not going to clarify it any further. I'm going to keep it kind of fuzzy, and we'll just bring the fuzziness into other systems, but more, that the definition doesn't super matter, but what we were really interested in is characterizing the behavior and then sort of understanding the sort of mechanisms of the behavior more than like any definitions. So yeah, great question that we're going to take further down into our framework of liquid and solid brains. Thanks, Santosh and Mike, for your questions. And so then, having thought through like the sort of what we get from the, like, having some really great stuff from the neural part of, or the solid part of the brain's world, some really great questions and some really great thinking tools, we go down into liquid brains. And this is from again this sort of broad overview paper from which the framework arises. There's sort of a lack of rigorous models to understand these, and sort of we're exploring that space. It's not nearly as developed as the connectomics of solid brains, for example, and what is the next steps in developing that. So we'll start with a computational lens as they oh, how sad. Parts of the slide did not load. All right. Hopefully the figures on the next page work. Anyway, let's see. There's a figure here. Weird. Oh no. Oh man. I don't know what's going on. Google Docs. Anyway. Well, there is a figure here. Darn it. What is happening? Bummer. Okay, well anyway, this paper's great. And the Google Docs application or the Google Drive. All right, I'll do that Santosh because this is a cool paper. One sec. Let's see. Come on. Okay, are you guys watching my Google Drive go real slowly right now? You're watching the pixels. Okay. Okay. I'm gonna have to get my desktop back to see. I hope you're seeing the liquid cellular automata now. Just have to get that chat out. Okay, great. Let's do this. Where'd that chat go? My God, the whole world's seeing my desktop. This is like worse than. All right. You're seeing your own chat, and yeah, you're seeing some liquid cellular automata, right? Cool. Okay. All right. This is a paper from my lab. I don't actually know this person, but I hope I meet him tomorrow. And these little guys are liquid cellular automata, so little computer agents that are just exist only in silicon imaginations, and this is totally liquid, right? This idea of liquid, right? And it's more of a molecular liquid than really like liquid, right? It's like if you're thinking about like on the molecular level, think air, not water, actually, even though we said liquid. And they are, in some, theoretical space, and they've got some information, and the black guys have, the information 1, and the white guys have information 0, and they're sharing information with each other. And this is at time 0. We sort of, evenly distribute the information as a starting condition, and the little edges are just the guys in their, I don't know what gender they are, the dudes, the bits in their vicinity that they can share information with, and then they form these little pods. And this is sort of like a first, like pretty intuitive result that if you don't let them move around at all, so S is the speed that the automata are moving, and yeah, automata are just right like the simplest theoretical instantiation of what we might call a computer, right? So this is time 0, and they if they collide, it doesn't really matter. Collision is also just sharing information. They're not like they're so sort of like abstract that they don't like they can't get hurt or anything with each other. They're only like they only exist in the context of being information sharing agents. So like like you can see they sort of overlap each other and they're just sharing information is like the only behavior we care about is sharing information. They're not competing or anything. And so if we let them not move, it takes a while for this is just like the purport. So what you're seeing in the wax is what we've done is have them give a consensus of just a bit of information. They all have to have the same consensus of a bit of information, a 1 bit, right? Like 0, 1, like simplest, sort of information. They just have to either converge on 0 or 1 is like the answer, quote, air quotes again, answer. And so this is speed is 0. They're not moving at all. And it turns out that it takes them a while, like, they're not all correct, or it takes them a while to get all correct, right? And then they get all correct pretty often once we let them move around, right? Pretty simple, like sort of intuitive result, right? We let them move around and share information, they do it faster and better than if we don't let them move around. But this is sort of, again, this is theoretically the fully liquid case, right? That doesn't actually happen in real life very often, right? We, like, if you imagine, like, in a city, for example, like, being able to interact with every other human in the city, like, that's, like, physically and so on so many other levels, like, a complete impossibility. Or even, like, an ant colony, which we'll get to, like, that the sort of ant colony version of this would be that if every other ant was physically and physiologically able to interact and get information from every other ant, which is like obviously absurd and sort of impossible, and sort of so it's it's a theoretical approximation on real life that is it's the spherical cow of a liquid brain, right? Spherical cow being this sort of idea that that starts like theoretical physicists when they go to think about a problem take something that is abstract and fuzzy in the real world like a cow, and hard to literally and figuratively get your arms around and just say, oh, pretend that it's a spherical cow, and then it's easier to write math about it and stuff like this. So this is the spherical cow definition of a liquid brain, and the authors sort of fully acknowledge that this is a spherical cow. And that, like, it is a mathematical treatment of the problem, but a complete mathematical treatment of the problem is challenging for a lot of reasons. That, like they say, like I just said, and they've said, like, the idea that all of these liquid cellular automata or ants or pick your favorite cognitive agent can actually be fully mixed like this is just not exist in the real world. And physical environments have boundaries that create edge effects and complicate mathematical predictions. And this paper talked about those two. It's to a certain degree, right? The sort of the space itself had these edge boundaries, so they dealt with that. But obviously, there's even more physical constraints in the real world than merely sort of the edge of your space. And as they say, physical topographies are complex and real systems are often asynchronous, which we'll get to those two in the real world in a minute. But they sort of leave the door wide open to say, like, this is sort of a first pass. These are spherical cows. This system in the simplest form behaves like we think it should, but that's not the complete picture. This is sort of like, we can get the signal of one sort of neuron, air quotes, talking to another, but that's not the whole connectome yet either. So that's a sort of the first pass, the first theoretical spherical cow pass at a liquid neural brain. And then now let's go to some real world liquid neural brains. And remembering Santosh and Meg's great question from the top of this framework. Let's go to let's take it down. And so we're asking, right, what happens if we, this is kind of a abstract, this spherical cows, right, abstract computational perspective of this system. What happens if we have even more of a cognitive lens on the system, not just, what does that look like? And so actually, I'm going to give you now some examples from the real world of liquid brains, realer world, so that we can sort of get it, like what the space of how this hasn't been thought about yet. So these are ants, right? So again, this canonical example of a liquid brain. These are the cutest, smallest little genus of ants that scientists love to work with, Pygnomyrmex. I can never say it right. But these folks, and this is very cute, they named these are two different colonies that they have been studying. One is named Beth. I think this colony is named Beth, and this colony is named like Gertz or something. So if you study ant colonies, you get to name your colonies, which there's actually some really cool theoretical work that like in theoretical biology that basically says that like, yeah, you should name your colony of ants and not your ants, because from a theoretical perspective, the individual is the colony and not the ant, as far as, metabolics and survival and, sort of what level of the system evolution is acting on. So yeah, this colony is Beth and it's going to live and die as Beth. Anyway, so these are ants, and you can see they're in this, sort of the real world example of our like, random square of space. And they've got a little entrance, entrance exit, so a little entrances, well, they're entrances and exits to the space, these little tubes. And they, these, like you can imagine this in their lab, and they just film these ants, and they get, these results. So what you're seeing are the paths of these ants, right? These little, and it looks, it looks kind of like a brain. And the red to blue, it's sort of a scale of the number of interactions. So that the dark blue here, like this ant, was sort of off in the corner, not really interacting with anyone, and this ant was sort of like the gatekeeper, right at the entrance to the nest, the laboratory nest, and shared a lot of information. So, if you're familiar with network science at all, or you've had a social circle ever, you're maybe starting to see what we call a heavy tail distribution already, just in. I want to go back actually, just in these photos, right? When we're seeing these patterns of red to blue, we're seeing this idea that sort of like there's some central nodes in this network that have then sort of periphery, right? We all have that friend that knows 50 other people. This person, or like knows way more people than we know. And when you say like, they're like, oh, I have a friend that does that, and oh, I have a friend that does that, right? And then we all have those friends that are, maybe more introverted, and we see them, but we don't have any friends in common with them. We just know them through this one thing, and we think they're great, but we don't, we just don't, and like maybe their network is connected over here. Anyway, so we call that this property of networks, where some nodes have a lot of connections and some nodes have less connections. We call that a heavy-tailed distribution, from like this graph, right? So it, it's sort of this is the heavy tail, right? That there's a few agents, people, whatever, that have a lot of connections, and then a lot that have a few connections, right? And that's also like if you watch YouTube, there's like the couple of content providers that have like all the views, right? And like TikTok has the same, I'm sure, distribution already. People are whatever studying it, and then a lot of folks with a few views. All right, so this is just a classic thing that it is observed a lot. And these graphs are the colors here are the weighted degrees of the ants. So degrees are just how many other guys am I connected to? So that's a degree, and then in graph science, that's a degree. And then the way they've built these graphs to just visualize this is that they use the equation of a spring, right? And then connect them with the sort of springiness of the spring is the amount of the weight of the degree. So how many neighbors you have is the springiness of the spring between you and everybody else. if that makes sense. So that's how come the guys with lots of neighbors, lots of high degree, are in the middle with lots of guys close to them. And then this is that heavy tail distribution, right? So we're starting to get see some patterns, see some things like the sort of like first pass stuff that we're expecting to see. We're seeing. So this is right. Remember that problem with liquid brains is that they've all of the problems with liquid brains is that the physical topographies are complex and they're often asynchronous. So these are problems we're like thinking about. And so here they've been able to take a small snippet of the physical topography of this system, and write down the network structure of part of the system, right? But and colonies are like big and complicated and have lots of physical topography, right? You can imagine that in the real world, these guys live in a colony that's quite bigger than this little box, and they have an immense complex architecture beyond this. So this isn't the full connectome. It's a snapshot of part of it, right? A physically constrained part of it, right? And actually, this lab has done some really cool work where they pour effectively concrete, but it's a little softer. It's like plaster into ant nests, and then take the nest out from it, and then they have a physical it's almost like a sculpture that the ants made. It's a concrete representation of the ant nest, and they look like coral reefs when you get them out of the dirt and unearth them. And they have been able to count the the degrees of the there's ants will have a chamber, right, and then a pathway away from that chamber, right. It's just a series. An ant tunnel is a series of chambers, and they count the degrees of those chambers, so the paths in and out. And that also has, a pretty normal. I forget exactly the pattern of that, but again, a small view into the physical topography of these networks and how they're architected. So very cool work. So that's again, we can sort of, because of the physical constrained nature of these things, it's hard to get the whole end to end system, how to even just get the data on it, because you'd have to have an x-ray on an ant colony or something. Who knows? Actually, maybe that's not a bad idea. And so then the last problem is this sort of temporal variation in these systems that moment to moment and over time they're changing a lot as well. And so this is some work that is able to take a non-physically constrained system that's that you can that you can see the whole system all at once in one moment, but it's very dynamic over time, and so it's hard to capture what's going on across a whole longer series of behaviors because every interaction is so complex. So these little dudes are fish, but they're just like the little liquid cellular automata. These are not real fish. Those were real ants. These are not real fish. These are sort of like spherical cow fish in a computational simulation. These are computational imaginary fish that have the sensory capabilities of real fish, more or less. And they are in the simulation. They're swarming, or they're schooling, in the ways that fish do. And these folks were looking at how is it that they might be connected to each other? What's the best way to think about how they, as a sort of network, right, of neural agents in a liquid brain? And this is this is the most liquid example, actually. That's I hadn't thought of that, but that's funny. This is they're also in fake liquid. They're fake fish and fake liquid. But how do we understand how they are sort of connected to each other from an information processing perspective, how are they perceiving each other and passing information between each other to achieve this intelligent behavior of schooling. So meta. Yeah, we crossed a line of like, yeah, meta a while ago. I'm a little confused myself. Yeah, and so you can see it's a little subtle, but right, this is, if I'm a fish, let's pretend that the way I'm able to think about my neighbors is I like, and these are these are also sort of straw man spherical cows of like, because you can kind of infer like empirically how fish are actually doing it because fish have eyes. But pretend fish don't have eyes, and somehow they have a weird mechanism where they can sense everybody in their, in a specific metric of, I can sense people x amount away from me. There's a arbitrary circle of radius arbitrary x, and I can sense other fish inside of that circle and not further. And that's how that's as much information as I get from the system. So obviously that's not how fish actually work, but just from a theoretical perspective, it's interesting to think about, right? And then this is topological, which is a little bit different, right? It's, again, assuming that sort of, rather than the circle is of this radius and I can see everybody in this circle, this is, I have a k, right, of I don't know why it's k and not x, but right, we have to use different variables for different things so we don't confuse ourselves. But yeah, say I have 7 nearest neighbors, and just by whoever is like the closest to me, the first 7 other fish that are the closest to me, those are the ones that I can see. So that's topological distance here, and like, let's pretend that again fish don't have eyes, and they that's how they're sensing their neighbors. And to be fair, there might be other there might be other things out there. There might be fish out there that swarm this way, that lives in the depths of the ocean. We just can't see them, right? Probably not. But this, my point is, this is one step closer to reality from like these liquid cellular automata. We're like thinking a little harder about like the abstract things in the real world, but we're not quite in the real world yet here either. But so, and then and again, this is a little less spherical of a cow, but still a spherical cow, because we want to be able to write like nice things about it mathematically, which they've done. These are very beautiful, mathematical thoughts about this. And so this is like a particular geometry of a way to draw these polygons around. It's a little bit like this metric thing, but a little more complicated geometrically. Again, a bit of a straw man to just think about like, how are they actually doing this? And theoretically, how might one go about doing a thing like the fish are doing? And then the interesting part of all these metrics, right? And then they get to visual, and they actually did some cool math about like the curve of a fish's eye, and then being able to sort of extrapolate from there what we can infer based on the angles of the curve of the fish's eye, like which fish it can actually see. So they've like, this paper is very cool, and if you go online, you can watch videos of the simulation in action, where these little triangle-y things are the visual areas of the fish, and you can kind of see right here that like this fish is blocking the visual view of this other fish. So that's, this is simulation-wise pretty close to the real world, and therefore pretty cool, I think. And so, yeah, with that this is some, it's a spherical fish eye that they wrote down the mathematics of. Very cool. And they show that the thing that's probably actually happening is the most effective thing, right? That like the visual distance has the most accuracy in the system. So yeah, but again, it's sort of constrained to a smaller margin of the it's not the whole picture either, but it's a lot closer. it's a narrow temporally, it's a narrow like time chunk of this network structure of a liquid brain, like. And you could probably write down in simulation what's happening temporally over this network structure of fish sensing each other across space and time. So yeah. Oh yeah, and so those are some real world examples, and then I have my own sort of straw man argument of to like further think about this question that Santosh and Mike have given us. Right, so this is closer to this. It's the beginning of a cognitive lens on a liquid brain system. And you might ask yourself, well, there's tons of or I ask myself anyways, there's tons of animal behavior happening, and folks have written down lots of network structures for animal social structures. And why are we thinking that isn't, why is that different from this cognitive lens that we're thinking of? And so these are some network social structures that were written down by some, animal behaviorists, ecologists, of the ways that disease goes through a social structure of animals. And I don't even know, I don't remember which animals it is. Kind of doesn't matter. The orange lines here are disease vectors, right? So if two of these guys are connected with an orange line, it means that they gave the disease to each other. And they're just looking at the way that the disease would spread through this population given these different network structures. And I would argue that this is a social lens. And so they're treating the system as a liquid aneural brain here, not a liquid neural brain. Which, if you want to get meta, maybe the virus is, if you think about this system, like these architectures from the perspective of the pathogen here, maybe it is a computational, so maybe it is a something we might classify as cognitive, that it's solving a problem in space and time to better infect the population. But that's like a little too meta. But as far as the sort of the agents in the system and how they're the animals themselves and how they're relating, the treatment of them here is that they're sort of aneural, that they're not cognitive agents in the system. They're just sort of being infected. And the nature of how they're related to each other is of interest to how they infect each other, but not like what they're actually achieving with their own cognitive abilities. So, but we know that animals do, like social groups have, cognitive abilities, and they work together, like these fish. So there are, and this group, especially folks in the sphere of V and Cousin, tend to take this cognitive lens of these systems. And the question is, can we get then? So not all animal behaviorists do, but some do. And so the question is, can we get to like writing down end to end the whole connectomics of a liquid brain? Can we understand what's happening in the whole ant colony, start to finish? Can we understand what's happening, start to finish temporally in any of these sorts of systems? And can that, fulfill all the great things that Santosh and Mike told us would happen? And when we use this cognitive lens, can we learn things that are, useful to swarm robotics and artificial intelligence in how to, construct architectures for distributed computation systems that achieve better results than achieve great results, make advances in swarm robotics, AI, and whatnot. So yeah, that's the question. Those are the thoughts. I hope that wasn't too meta for you, too crazy, but those are just some thoughts about the connectomics of liquid brains. And I'll just hang out if you want to talk more, want to have any questions. And I hope that made sense. I hope I answered the question about, that liquid is a metaphorical. We're thinking of liquid particles of air more than really water. Yeah. I keep telling Melanie they're not liquid, they're semi-solid, but yeah. Shape-shifting. I like that. Malleable. Yeah. It's sort of at the moment, it's sort of a straw man to just like bounce ideas off of, like, yeah, it moves. I mean, it doesn't move all the way, but yeah. And in the sort of the whole, in the liquid brain, solid brains issue, there's also like statistical physics where they again treat the system as completely well mixed, and I don't even know if they say that, like, yeah, that assumption is kind of silly. They're just like, we're theoretical physicists, it's fully mixed, it's fine. It's a good paper. It's just what happens if you do those dynamics over a network structure. Yeah. So if anybody wants to find me, of course, solid brains are always changing. Yeah. Yes, right? That, like, it's solid and liquid are both maybe not the right term, because, yeah, like neuroplasticity, like neurons do die, and you can get new ones, and yeah, they are, yeah. And the yeah. It's a it's a continuum, not a binary, is I think the way I'm thinking about it now. And nothing, yeah, only do the like purely solid and purely liquid, they only occur in our simulations. Yeah, but if anybody needs me, I'll be making some liquid cellular automata, like seeing what happens when you give them a small world architecture to work with. So, yeah, spherical fish cow cellular automata. Cool. Well, thanks for tuning in, everyone. Tune in, not next Friday, but the following Friday. I don't know what's going to happen, but it's probably going to be exciting. It will be season 2, episode 4 of Proteus Television. Yes, and it will probably be interesting. We'll see you there. And then at 3 o'clock Eastern, as you know, 1 o'clock Mountain. All right. And I recorded this. I think so. Okay. Thanks, everybody.